Hi, so welcoming back to my channel. Here we look at RNA velocity model for single cell RNA. So in this we'll just look at how the RNA velocity model idea came into the picture when I was researching on my PhD content, but it's more of like it's my daily research, and it's a very important topic that I came to know about, and I will gaze through all the information. But the entire code application of the demo that you will see, I will show it in a proper manner, step by step, in subsequent videos that I intend to create. It would be a series where you get the information of it. So what is RNA velocity model? So RNA velocity model for single cell RNA sequencing is a computational method that predicts the future state of individual cells. So when we are studying say a current disease, or say if you want to work on a particular drug, you want to think of a situation like how a particular application reacts when we add some other extra content to it so that it changes. So in general what it happens, it breaks down. So with this RNA velocity analysis, we are able to see what the future state of the individual cells can be. So it is done by the process of analyzing the ratios of unspliced and the spliced mRNA transcripts in each cell that we are working on. Okay, so the key concepts over here. So three particular key concepts when you are studying the RNA velocity. So it is splicing, unspliced versus spliced mRNA, RNA velocity as well. So splicing, the process of removing introns, that is non-coding regions from pre-mRNA to create mature RNA, which is then used as a template for protein synthesis. So unspliced versus spliced mRNA. Unspliced mRNA indicates recent gene activation if there are changes. So when you are studying cancer, we need to see like if there had been changes or not. So with this velocity pattern, we can study that. While spliced mRNA reflects the current state of gene expression. So we can compare both of them. RNA velocity, the rate of change of expression over time inferred from the ratio of unspliced to spliced mRNA. Okay. Now how it works? So single cell RNA sequencing, this technique captures the mRNA content of individual cells. You will see also in the demo itself, providing a snapshot of gene expression. So when we are studying a gene, we have to entirely study how it works, what are the patterns for that. We need to go through it. Unspliced and spliced mRNA quantification. The single cell RNA sequential data is used to quantify the abundance of both unspliced and spliced mRNA transcript for each gene in every cell. RNA velocity calculation. So the RNA velocity model uses mathematical algorithms to estimate the rate of change of gene expressions based on the unspliced to spliced mRNA ratio. And as I was studying the DINVerse for a longer period of time, so if we just go back, I will just open up DINVerse. So here is DINVerse. Okay. So DINVerse has a very high trajectory interference details. It has a collection of 50 plus trajectory information method with common interface. But the entire application that you see is like it's created in R. We can create wrapper over R for Python and use it, but it would be very difficult for analyzing. So I thought of creating a tool or like a complete analysis application that we can infer this trajectory inferences for different kind of genes that you see. So DINVerse came into the picture. So what I did was I studied it in R, and I was trying to find out like if there is something that can help me build the application. So as I was researching, so this particular library came into the picture. That is it is called the scVelo, or we can say that it's a library for RNA velocity generalized through dynamic modeling. So scVelo or scVelo is a scalable toolkit for RNA velocity analysis in single cells. Okay, so I took this as a starting point and identified what are the things that I can do with it, and converted into a complete, what you can say, a toolkit. Okay, so we'll see that in action. Actually the entire code is currently written in Streamlit, we can completely use it as a application and can get it as like an Android app, or say, or you can have an iOS app for that, so that we can analyze the entire data set of it. Okay, so let's go back. So how it works, we have given the information of it, the cell trajectory. Now applications. So for the applications, we have developmental biology, understanding the dynamic process of cell differentiation and lineage commitment through development. Disease research, identifying the cellular and the molecular mechanism underlying disease progression and response to treatment. Very important. The second part that you see over here, that is disease research, identifying the cellular and the molecular mechanism underlying disease progression and response to treatment, is very important part. Now we have regenerative medicine, discovering new therapeutic targets for regenerative therapies. So these are the application areas for RNA-velocity mechanism. So let's see it in action right now. So the entire code that you see is being written in Python. Currently, I'm not going through the entire code itself because I will explain it step by step in a later video, but I will see the demo of it, how it works. Now it is completely here hosted. You see the trajectory patterns has been shown over here, and you can select between RNA velocity, velocity graph, and gene expression. So if I just change from RNA velocity to velocity graph, let me just give you what exactly this example contains. So this example contains a dataset from scVelo that I have already differentiated and have loaded. So from there, what I did was I went through the application. At scVelo has different kind of datasets already pre-built. So I started building the blocks of the entire application using the available data sets that are there, and I chose one because it has a specific pattern that needs to be added so that you can do the RNA velocity analysis. Otherwise, your entire application or the work that you're doing will not be according to the liking, and there would be errors in the program. So you need to be very selective in terms of the data sets that you use. Okay, so currently, as you see, as you go back, it's a sling application here. You can see I've plotted the RNA velocity, then there's a velocity graph, then we have the a slingshot for trajectory inference, and finally, if you get more information on that, you can also study different things. So if I change from RNA velocity to only velocity graph over here, so you will see that it goes through changes. And now if I go back to the application, it tries to analyze the data. So when we are using a data set in terms of analyzing the bioinformatics data, it is essentially known as AND data that we are working on. And currently it creates the velocities. You'll see that it goes through using or computing the velocity graph of the application of 1 to 8 cores, already a data set that's been given, and you'll see the result very soon. Just wait for it. So it meticulously goes through all the steps, as you see, and there are different algorithm patterns being collected, and finally the entire thing is plotted like this. So you have a complete velocity patterns for these clusters, and we are for this data set, probably I am looking at the lung information, I guess, of endocrine or something like that data set that I've used. Probably I will just go through it when I create it from scratch and show it back to you, and you have the velocity graph also shown. So this is a complete application in bioinformatics that can be used in pharma as a tool that people can use it to excel in different patterns and also do external research in terms of the bioinformatic technologies that are evolving. Thank you, and keep tuned, keep in touch with me. As you see the videos coming in, you will see a lot of content that is related to this, and also you'll see another application that I've built using Kepler Mapper in future episodes. Thank you.